Welcome to tonight's masterclass. My name is Robin Young. I'm the publisher of Orthopedics This Week, and it's my privilege to introduce your moderator and speakers for tonight's masterclass. We truly have an exceptional masterclass tonight, and they'll be discussing an evolution, perhaps even a revolution, in PEEK implants, and that is Curitiva's 3D printed porous PEEK. Now, this is a particularly timely subject. First of all, there's the advent of 3D printing, which has really transformed implant designs in the practice of spine and neurosurgery. Also, the FDA recently cleared for the first time the first 3D printed porous PEEK implant, which was innovated by, of course, Curiteva. And Curitiva's ability to create an architecture for its porous PEEK implant has not been obtainable by any other company or method and delivers unique and beneficial properties to patients and therefore orthopedic and spine surgeon practices. So, explaining all of this to you and really creating an outstanding program and masterclass tonight is really one of the best master faculties we've ever been able to put together for one of our classes. We start with Dr. Alex Vaccaro. Dr. Vaccaro is the president of the Rothman Institute and chairman of the Department of Orthopedic Surgery. He's co-director of the Regional Spine Cord Injury Center of Delaware Valley and co-director of the Spine Fellowship Program at Philadelphia's Thomas Jefferson University Hospital. And that's where he instructs current fellows and residents on the diagnosis and treatment of various spine problems and disorders. Also joining us this evening is Dr. Kevin Foley. He is the chairman of the Sims Murphy Clinic, director of the Spine Fellowship Program for the University of Tennessee Department of Neurosurgery, and chairman of the board and medical director for the Medical Education and Research Institute. He's also professor in the departments of neurosurgery and orthopedic surgery and biomedical engineering at the University of Tennessee Health Science Center in Memphis, Tennessee. Also joining us is Dr. Barbara Boyan, PhD. She's the executive director of the Institute for Engineering and Medicine at Virginia Commonwealth University. She is also the professor of biomedical engineering, where she holds the Alice T. and William H. Goodwin Jr. Chair. All three have been true thought leaders and indeed giants upon whose shoulders we all stand in the practice of spine and orthopedic care. So with that, I will now introduce our moderator, Dr. Eric Irby, PhD, who during his 30-year career played a critical role in developing and commercializing biologics, synthetic biomaterials, and medical devices in orthopedic and spine, and he holds 54 issued patents. He's been on the podium a lot, and currently he is the chief scientific officer, Dr. Irby, at Curiteva. To our audience, do make use of that Q&A button. We love to get your questions. It's at the bottom of your screen. And now I will hand the program over to my good friend, Eric Irby. Dr. Irby, the program is yours.
Thank you, Robin, for that wonderful introduction. I am indeed honored and privileged to be representing this conversation with this illustrious faculty. But before we get into the conversation, I'd like to begin by echoing your sentiments, Robin, about this being truly a revolutionary technology, an enabling technology that's not achieved and hasn't been achieved any other way. The 3D printing of a PEEK trabecular lattice, to me, is a culmination of decades of work. There's a lot of effort, myself included, going back several decades, looking at trying to make PEEK bioactive to overcome its Achilles heel of creating fibrous tissue because of its hydrophobicity, despite some of its wonderful properties. This approach of 3D printing, as we will learn tonight, gives you structures you just can't get any other way. And I've not seen truly any other technology out there. Just saying you have porous PEEK is not the same as 3D printing a trabecular lattice, as you will see tonight. So again, I'm honored and privileged to present this. Again, personally, looking at decades of material science and R&D, trying to develop better products for surgeons to deliver better outcomes for their patients. And I think you'll see that tonight with this 3D printed PEEK trabecular architecture. What we want to really get into is describing what we have here, which is a really novel biomaterial and a novel medical device that meets a lot of needs that surgeons have been looking for. We've gotten really close, but finally we have arrived. And we were truly inspired with our Inspire Porous Peak 3D printed implant. And part of what we wanted to achieve was full interconnected porosity. Having developed bone grafts in the past, the body likes pore structures that it can grow bone into to provide stability over time. Part of what has achieved this with our interbody implant is HA fused technology with our partners at ProMimic. And this delivers a fully osseointegrated implant in ways that you typically don't see with Peak. These are the attributes. And in the Inspire Porous Peak, we often refer to it or think about it as synthetic trabecular bone. It is 3D printed, and we'll talk a little bit about the differences of different 3D printed constructs. This is done by fused filament fabrication. And we do create, as Robin said in his wonderful introduction, novel architectures you just can't get any other way. You can't get them by compression molding or subtractive manufacturing machining. Everybody knows Peak has some ideal inherent properties: radiolucency, modulus, very close to cancellous bone. We've matched that in our engineered structure and also provided superior strength and toughness. That's very important when you start to add porosity into an interbody implant. Let me ask Dr. Vaccaro to weigh in on, as he looks for an interbody implant material, what are the things that you look for in considering whether to use titanium, whether to use Peak, or to use allograft bone?
Sure. I've been operating now for over 25 years. I was a resident in the 80s. I did my fellowship in 1992. And I learned early, especially when you're a younger surgeon, you take care of a lot of elderly patients. And as we know, society is getting older. And the biggest nightmare you have is when you see a migration of a metallic implant through osteoporotic bone. You can see that with a pedicle screw pulling out of vertebral body, or you can see with an interbody spacer subsiding. So we learned early to try to match the modulus of whatever implant you are with the host bone. So if I'm dealing with an osteoporotic individual, I would like to have an osteoporotic allograft bone if possible. And a lot of times I use a patient's own autologous bone graft. I, in the vast majority of my cases, use Peak. And I use Peak for several reasons. I use Peak because I can see through it. I can see a bone which is placed in it is healing. I love the modulus. It tends not to subside if I do an appropriate end plate preparation and I have the appropriate surface area. And now that the technologies have changed, and I really was an advocate of consulting with Curiteva because they came up with a biomaterial, a sort of 3D printed lattice that had a roughened surface, had the appropriate porosity, had the appropriate pore strength, and an early clinical usage. So great outcomes so far. So it's everything I want in an implant. I want an implant that I could see my growth, my graft healing. Now we know that titanium has a great track record of healing, but this is an opportunity for you to have sort of what I call an invisible implant that you can see and be sure that you have a solid fusion. So that's why I was drawn to Peak.
I'd like to ask Kevin his thoughts about material selection in terms of what's available for interbody implants. Certainly allograft bone, titanium, and Peak. Kevin, if you could comment on your thoughts about material selection.
Sure, Eric. So when I'm looking at choices for interbody implant material, I'm thinking about a number of factors. I'm thinking about the biology. I'm thinking about the biomechanical requirements. I'm thinking about bone radiography, and I'm also thinking about availability. So from a standpoint of modulus, titanium has a modulus, solid titanium, well beyond bone, right? Well, cancellous bone has a modulus around a gigapascal, solid titanium, 100 gigapascals, no match. And we can get into discussion later about how you can modify the modulus of titanium, but there's a mismatch there. titanium is bone friendly. We've all seen bone grow through titanium and onto titanium surfaces. Titanium is not radiographically friendly. So for my other criterion of the bone radiography, I can't detect diffusion very well through a piece of titanium, right? And certainly not on plain radiography, not without bone outside the piece of titanium, for example. So it's just not ideal. For PEEK, a solid piece of PEEK has decent biomechanics. Load bearing is fine. The modulus is around 3 gigapascals, a little bit beyond cancellous bone, but not bad. The radiography is ideal. I can watch with plain films a fusion occur through a piece of solid PEEK. The downside of solid PEEK is that it's not biologically friendly, right? The bone will not adhere to or grow into that surface or attach to it as it will to a piece of titanium. So we're left with choices that all have some issues. I think that's where taking PEEK and 3D printing it with porosity, for me at least, it represents a breakthrough, because that material checks all of the boxes that I need checked to find that ideal interbody implant. The 3D printed porous PEEK is very friendly biomechanically. It can load bear quite well. It also has a modulus that's almost identical to cancellous bone. So it checks my biomechanical boxes. Biologically, the Inspire porous PEEK not only has the porosity with pores that match Mother Nature, both in shape and in size, by the way, but in addition, there is chemically bonded hydroxyapatite within the porous PEEK, checking the boxes for the requirements for an ideal interbody material. I think the Inspire porous PEEK is difficult to match when compared to the other materials.
That interconnected porosity does indeed yield full osseointegration. And again, not words you typically think about in the same sentence with PEEK, but hopefully we'll have some compelling information for that. So how do we accomplish this? Well, Curiteva, you may not have heard of us. We're in Rocket City, Huntsville, Alabama, known for technology and developing world-class products. We're a world-class quality organization, ISO 13485, as well as MDSAP certified and registered with the FDA. In terms of getting to a 3D printed interbody implant, there's a lot of individuals, really smart folks working on that. And what made that happen for us was the acquisition of Fossil Labs, a company that was founded by Todd Reith, who is a unique individual who brought a skill set together and a focus to actually program, build hardware and firmware, as well as programming the software to build a 3D printer ideally tailored for printing of PEEK implants. We selected diffuse filament because selective laser sintering of powders is fraught with significant problems in terms of consistency, high scrap rates. What we have come down to is a very scalable apparatus, a piece of equipment that is very cost effective, very translatable. And so what was very critical in our clearance process with the FDA was helping to define and understand the critical build parameters, and then showing the IQ-OQ-PQ, which is a process qualification, showing that the machine is indeed making and printing what we design. And the first part is the same as the thousandth part. And I think that becomes very important in terms of calibration, control, monitoring, and recording. And in getting our clearances, though it was on the tail end of COVID, it took 18 months. which is an extremely long time. And in that process, FDA learned about the control and the precision that we had in this equipment. And that's what we believe led to our clearance. So that occurred early this year in February. And within 5 weeks, we were already starting clinical implantations. And we printed this structure, which you can see here as this series of images goes from the implant itself to the macro scale down to the micron and nano scale, where this is what the body sees when the cells come in there and touch the surface. So, Alex, I know you're an advocate of this premise of structure drives biology. As a surgeon, if you could kind of explain why looking for these things is important, and in terms of the outcomes of getting bony ingrowth, why that's important for you in an interbody implant.
So the things that you want in an interbody implant. So we've already talked about the fact that I like it to be invisible. I like to be able to see the bone that grows through it. Other things, I want to make sure it's stable. So I need to have a roughened surface. So if you look at the macro structure and you look at the micro coating, we see two things. We see that HA permeates throughout the entire implant. It's just not on the surface. It's not going to be flaked off when you place the interbody spacer. It has sort of the acicular whiskers of the HA on the surface. So the bone cells and whatever cells like to oppose to the surface, we call it osteopromotive. So we like the biology. We also like the fact that the pore size is appropriate. I think it's on average 250 microns because bone loves to form in that type of environment. And if you look at the histological samples from the sheet model, you won't see the fibrous tissue that you saw in the older peak implants that didn't have this rough surface. So those are the reasons why I like it. I like the cellular level, the fact that bone loves the surface. I like the macro level. I mean, you have the struts that give it stability. You have the modulus that prevents subsidence. And you also have the porosity and the interconnectivity that allows bone to go through it. So it has all the things that you want in an interbody spacer, plus it's invisible. And I'm coming back to that fact that I could see through it. I'm confident that healing was happening. And I'm not criticizing titanium, because we know that titanium has all those features, but this is an invisible implant that we didn't have before. PEEK used to be hydrophobic, and now we have an implant that's hydrophilic. We have an implant that cells like, rather than forming an apposition of fibrous tissue. So I think Curiteva hit a home run with this implant technology and biomaterial.
So let's talk about that a little bit in terms of the enabling nature of 3D printing allows us to print what is effectively a synthetic trabecular bone. In the images you see here, we have a macro image of the implant itself. And then at kind of a macro level, optical microscopy, you can see the interconnected struts that looks kind of like trabecular bone. They're interconnected porosity. And in the micro and nano scanning electron micrographs here, you can see the surface roughness. And then at the nano level, you see the hydroxyapatite crystals that really drive that cellular biologic machinery.
You know, all 3D printing is not the same, right? When you're taking a powder and centering it, essentially melting it layer by layer into shape, you're counting on the fact, for example, that all of your powder gets centered. Well, we know that doesn't necessarily happen. There's powder that can remain despite that centering process. When it comes to the manufacturing process that the Curiteva porous PEEK goes through, there is no powder there, so that's not an issue, and the process itself results in a porous structure that is almost identical to natural bone. It's through porous, so the pores aren't just on the surface as exists for some of the competitive products out there. And the shape of the pores and the size of the pores, as I mentioned just a few minutes ago, are designed to mimic Mother Nature. So the pores within the Curiteva porous peak are diamond in shape, and they are in that size distribution that you have listed here, Eric, of 100 to 600 microns, just as happens with Mother Nature. And so the material that Curitiva's printing is much more similar to a piece of human bone than any of the other materials that are currently being 3D printed.
So it's important to differentiate, because there's some other peak materials out there that have calcium phosphate, hydroxyapatite incorporated into the peak, which traditionally makes it a little stiffer and a little less strong in tensile strength. What we did is applied this nano coating to the surface, again, the entire surface, in the pores on the outer surface where it's solid. And this is essentially on the right what the body sees, which are, again, whiskers, needle-like, hydroxyapatite. That makes that surface hydrophilic, which is important for bone formation and apposition. And again, it's hydroxyapatite, so that's a familiar chemistry that the body sees. So at this point, I would like to maybe ask Dr. Boyan to weigh in on some of these observations. Again, this is the early structural elements and things that we're doing here with Peak that maybe have not been done before. Barbara, I don't know if you have some thoughts about this.
Thank you, Eric. Yeah, I have to say that the first time you showed me this structure, I got very excited, because it is the first thing that you think of when you look at it is trabecular bone. I have never seen polymer printed this way. There are a lot of claims about porous polymer implants that have holes in them for sure. But in our own lab, when we've looked at the way that these kinds of voids are created in polymer printing, what we have noticed is that irregular shapes are what bone likes, and shapes that mimic the natural contours of bone, they particularly like. So it was really a pleasant surprise to see something that could visually, I could look at it and say, that looks like bone, without worrying about it. Kind of looks like bone, it sort of looks like bone, it looks like bone. And I think that's part of why bone grows through it the way that it does. It's a comfortable, I know as a biologist, I'm always anthropomorphizing everything, but it does look like something like home that bone would find comfortable. And I also think the surface structure is part of that as the cells migrate in, they are comfortable migrating, and they do migrate all the way through. And bone is growing in fact, by creeping substitution, but it's growing through. And that's something that you often don't see. It's not that it grows part way and stops. It actually makes it all the way through. And it's an intriguing way of thinking. It's not unique, the idea that there be something that is structurally like bone, but to actually achieve it in a polymer was very exciting. Back to you, Eric.
So in terms of that structure, since we can print whatever we want, the diamond TPMS is the structure that we selected, very similar to trabecular bone you see on this scanning electron micrograph on the right, compared to our structure here on the left. And one of the things that's important is the radius of these struts, that they dissipate the strain energy and the load that the lattice is feeling. And so that's very bone-like, very, very important attribute. And so you see a lot of implants out there that try to target this 1 gigapascal stiffness. Titanium is extremely stiff, and despite putting a bunch of pores in it, it's still a pretty stiff material. So in terms of matching this modulus and the invisibility and some of these other attributes, perhaps you could comment on thinking about that load sharing and the bone graft itself, the importance of Wolff's law, and actually allowing that bone graft to be loaded physiologically.
So when you do an interbody implant, you have several things you have to keep in mind. Number one, you don't want to disrupt the subchondral surface. You want to sort of make it smooth. You want to make it contour to whatever interbody space you're placing. Your implant has to have a surface area where it covers as much of the supporting bone architecture as possible. So you can take a 3D printed PEEK substance, and I'm sure you don't even need to have an opening in the middle for bone graft. I'm sure with surface characteristics such as the HA, the interconnectivity, the fact that HA goes throughout the entire implant, that you could just have a pure PEEK cage with the appropriate lattice with a diamond shape to allow the cells to come in. But this implant, Inspire, has that ability to allow you to place bone, whatever substance you want to place inside. Some people use local bones, some people use a demineralized bone mix, and so forth. So for me, I want a large implant that covers much surface area as possible. I want the modulus to match the bone. And we say, what's the perfect modulus? It's 1 gigapascals. That's what you really want. I want something that if it begins to subside, it doesn't catastrophically collapse and fail. And I want something that I can get a nice x-ray that's sort of radiolucent. So that's what I look for from a biomechanical perspective at an interbody implant.
Alas, we must also show strength. And these are some of the tests that FDA wanted us to show. And this is very important because at a minimum, we need to show compressive strength and fatigue strength, which we did following the ASTM test methodology. We tested our smallest implant. You can see here in the lower left is the image of the implant that withstood 25,000 newtons. Now, I can tell you that that's the limit of the load cell. And at that point, we stopped the testing. And this is what occurred, about a 3.5 millimeter deformation. On the fatigue sample, there's 5 million cycles at 1,500 newtons, representing 2 years of ambulation. This is the sample, no fractures, no particulate generation. Again, all compliant with the FDA guidance in ASTM 2077. Compared to traditional PEEK, and in defense of them, they have very large graft windows and perhaps not the engineered lattice structure that we do. But a lot of these implants will fail at a fraction of the load that we failed at. And they fail catastrophically, meaning they completely buckle. And that's certainly something you wouldn't want to see. Not that that happens frequently, but we're significantly higher than that, given the human vertebra can withstand up to 6,000 newtons. More importantly, FDA, and I think this arose from their concern of other materials, in particular compression molded PEEK that had surface porosity and lost height and material upon impaction. They wanted to look at our impact toughness, impact resistance, as well as they wanted us to repeat our fatigue testing. And they threw a little twist in, which is with and without the marker. There was a belief that maybe the marker was serving the titanium marker, which in this image you can see there's 2 here in the front, 1 in the back, wanting to make sure that wasn't acting as rebar in some way. And indeed it was not. We were pleasantly surprised when we did our pre and post testing, not only in dynamic loading, but in impaction. We went as high as 5 times physiologic load. And the most we could ever get was about a 30 micron compression of the overall height. And we never saw anything much more than that. One was 40 microns. So very insignificant, no meaningful alteration or change. That gave us confidence. How important is that to you as a clinician to have an impact resistant and tough implant?
So it's essential. The biomechanical requirements of these implants, as you've mentioned, go beyond their need to sustain loads, both static and cyclic. Once you've placed them, they have to be able to resist the impact loads that are placed upon them when you insert them. And so the unique structure of the 3D printed Inspire implant allows that combination of an ideal modulus with tremendous strength, as you've just spoken about. One can impact these implants and not deform them. They resist that. I think part of that is your strain-induced crystallinity. I think part of that is the fact that all of these pores are interconnected, Eric. If I take a solid material and I just produce porosity on one surface or another surface, theoretically, I've weakened that surface, right? If instead I produce pores by this interconnected trellis, right? All of these interconnections acting together to resist the load, I've got a naturally stronger structure. That's why bone is like it is. And then I'd add another couple of observations here. And some of this comes from my work clinically, which wasn't apparent to me when I first saw this material. Because the surface that is adjacent the bone, so your superior and inferior load-bearing surfaces of your implant have that same porous structure. These interbody implants, at least in my experience, interlock with the bone early. In other words, you don't need ridges on this implant or anti-back out features, because that structure grips the bone.
Now, Barbara, this is because of our perceptions about PEEK, and in the past, most traditional PEEK it elicits this inflammatory fibrous encapsulation. We didn't see that, and there's some reasons for that, but we wanted to generate a burden proof. So I'll turn it over to you to describe the cellular work that you did for us.
Yeah, thanks. The experiment that we set up was similar to experiments that we've done with other materials. We put the cells, in these cases, we use human bone marrow stromal cells. We culture them on the surfaces, and we always have a control, which is tissue culture polystyrene. That's the TCPS there you see on the slide. When we cultured these cells on these surfaces, what we found was something really surprising. On the porous PEEK with the HA coating, there was a marked increase in VEGF. That's the vascular endothelial growth factor. What makes that so important is that bone has to grow through a lot of pores, and those pores were not, it wasn't like a tunnel straight shot. Bone is growing in and through, and doing that, it must have a vasculature that is happening at the same time. What I thought was fascinating was that these cells are producing the factor that would encourage that vascular ingrowth, and that's important for the nutrition of the bone as it's growing. The other thing that we found, which was really exciting, is that when we cultured immature monocytes on these surfaces and let them differentiate into macrophages, we found a really remarkable piece of information, which was perhaps the most important thing that is happening here, is that the macrophages are differentiating along a pathway that is called the M2 phenotype. The M2 phenotype is associated with bone regeneration, with healing, with I'm trying to figure a way to say it that's not scientific so that you get the excitement that I feel. But it is associated with regeneration. An M1 phenotype is associated with a chronic immune response. Now, that doesn't mean that cells in the M1 phenotype aren't important. They are, because they set up the whole process. But if we don't see that transition to M2, then we don't see regeneration. And what was remarkable about what we saw here was these cells are definitely going down an M2 phenotype pathway, which is probably explaining why that bone is able to grow through. Not only is it getting information so that the vasculature can grow, and that's important because the immature cells are migrating in with the vasculature, but they are also getting information in an immune sense that the environment should be one that is positive about regeneration. And the remarkable things that we saw were that TGF-beta was upregulated, IL-4 was upregulated, IL-10 was upregulated. IL-10 is always associated with a very healthy M2 phenotype. But even more importantly, the chronic inflammatory cytokines were downregulated, IL-1, IL-6, and TNF-alpha. Now, better than this, you can't get. It made me a believer You can get bone to grow a lot of different ways. It's not all about cells becoming osteoblasts. They become the bone has to form in a bone conducive environment. And this surface is certainly setting up the immune response to be conducive to good regeneration of bone.
So, Barbara, your insight into that was very, very significant for us. And if you recall, there, because of your and my past, a little bit of arm twisting, maybe to think that we might even see anything interesting in this endeavor. And indeed, we did.
Yes.
And these terms, immunomodulation, osteogenic potential, as surgeons are looking to understand these mechanisms more clearly, you're hearing more talk about these with bone grafts. And in fairness, now I'm going to move to additional study, and I'm going to ask for your feedback on some of this histology here in just a second. But in fairness, what won my ability to gain your attention was showing you some of this early data from Professor Walsh at University of New South Wales in Australia. And indeed, we had done a bone implantation study with these sheep. We picked older sheep so that it would be a more challenging bone formation model. We looked at not only cortical bone, but cancellous bone. And these images you see are cortical bone histology at 12 weeks. So a reasonably early time period for a human, not so much for a sheep. These are the comparative images. On the upper left, you see a histology section from a 4-week-old implant. You can see the new bone starting to grow into that pore structure. And on the right, you see now a 12-week cortical bone, and that bone has grown completely across. What is noteworthy is the absence of any of the dense fibrous tissue that typically you might see with a PEEK implant, a blow up of that histologic interface. And what's nice, again, no dense fibrous tissue. You can see in these lower images, the native bone here. This is new bone growing in. Same down here. On the right, you can see native bone and new bone growing into that space. And filling it up quite nicely, and you get a sense of this this intermingling and a biologic not only friendliness, not being rejected by fibrous tissue, but this bone looks indeed. I'm going to show one more image, and I'd like you to weigh in, Barbara. This is a stereo microscopic image, so it's the same stain, but the color looks a little different. And when we zoom in on this, I've labeled a few things. And what's interesting to me is the point you were just making, Barbara, about the VEGF and some of these other growth factors and proteins that are being expressed. You see blood vessel formation, you see bone formation, you see structure to the bone. And again, this is, we might call it beautiful bone, but it shows that that bone is viable and physiologic and nicely osseointegrated. I'd love for you to comment on some of this, Barbara.
Well, you know that I have one of these slides on my bookcase, so I can see it every day. I never thought that I would see a PEEK product that made me think that it was worth putting in bone. So for me to choke it out, Eric, it's a nice product, and it's clear that bone is comfortable integrating in with it. The mechanism involved is not the same mechanism that is operative with titanium, and we shouldn't expect it to be. PEEK is not titanium. So that it is doing this and doing it by a different pathway is perfectly fine with me. It's quite exciting.
I think, Barbara, that's a wonderful statement, and I couldn't agree more. And maybe that's something that a few researchers could look into a little deeper and figure out what's exactly going on with that bone formation. Okay, so transitioning our discussion to the clinical space, I'd like to discuss the first surgeries that have occurred with the Inspire implant. There are 22 surgeons. It's only been a little over 5 months since we did the first implantation in April, and we've got over 200 patients already. And I think that really speaks to the excitement of surgeons wanting to get their hands on a product such as this. And in those 200 cases, there are 377 levels. So you can do the math. They're not all 1 and 2 level. We have several 3, 4 level, and quite frankly, inspirational, if you will, 5 level that shows, again, the surgeon's confidence. And we're going to hear about that in terms of what this looks like. And so going forward, we will talk later about a clinical registry where we're going to collect this information and get these results published. And so without further ado, Kevin, I'd like to lead in with some of your first work, pioneering cases, I believe, a week after we started Implant. So Kevin, could you describe to us your early experience with the Inspire implant?
Sure. So these are obviously radiographs from a 2-level anterior cervical discectomy and fusion. Pretty routine case, but I'll highlight, Eric, some of the features of the Inspire porous peak that I think are helpful and unique. So on the left there, that's a picture I took with my iPhone of one of the implants before I placed it. And my favorite biologic, so to speak, for interbody work is autograft, but local autograft. So I save the grindings from my high-speed drill work and use that as my biologic material. And so what you see on that left side is the powdered autograft from my decompression press fit into that piece of porous peak. And if you flipped it over, the other side would look identical. And so there's a small central cavity in the current Inspire implant. Hard to see here because I've filled everything with bone, but you literally can take the material I use, that powdered autograft, and press it right through that piece of porous peak. Some things to look at here. You see the bone at 30 days within the implant. I think you see it quite well at 3 months. And I'd note that the central cavity in these Inspire interbody implants is smaller. And yet if you look at that implant, you'll see bone throughout. That implies to me that bone is growing through, Eric, the entire interbody implant. I'd also point out that you don't see significant subsidence here. If you look at the bone vertebral body interface, it looks good. It looks identical at 3 months to the way it looks at 30 days. So from my perspective, I'm seeing the things I like to see with an interbody implant. I'm seeing these things behave biomechanically. They're clearly load bearing and not deforming. They're clearly friendly biomechanically in terms of that modulus to those adjacent vertebrae. But I'm able to see the bone quite easily within these interbody implants with plain radiographs. So all of those things are features that appeal to me.
Kevin, you had mentioned to me that and earlier you had commented about the tactile feeling when you inserted that implant, that the tendency is for it not to want to move once it's in there. And that's indeed in part due to the kind of roughness of that interface. You want to comment on that?
Yeah, well, I learned that one the hard way, Eric. I like symmetry in the OR. I think it's beauty. So I like those little markers to be parallel to each other on those lateral radiographs.
Yes.
And so I had put one of these implants in, And frankly, it was a little askew. I could see 2 markers on the front instead of 1. It doesn't appeal to me. I thought, well, I'll just take the inserter and move this over a little bit. I couldn't do it. I had to grab a coker and to rotate the interbody implant. And so it speaks to that phenomenon I talked about earlier. These implants have a surface that interlocks with those adjacent bony surfaces as you insert them.
Well, that was very important feedback. Now we're going to talk about some of the cervical cases from Dr. Dreyer had submitted these. And one of the things that we were eager to see was, again, that bone growing shorter distances into the porosity versus the time it might take for bone to grow through the entire graft window. Could take certainly 3 months, if not longer. But with this pore structure, we were really looking for bone growing into the porous implant. So maybe you could comment on a couple of CTs we have here on these 2 cases.
Sure. Well, one of the things you can see here, for example, is the size of that graft window. Look at that third axial CT from the left. The denser bone is within that graft window. But if you look at the 3-month sagittal scan, the second from the left, you can see the slight artifact from the titanium pins. But you'll notice that there's bone not only through that graft window, but also growing through that porous peak.
Yes, that was quite exciting for us when we saw that. Where the case in the humans, we now have more compelling evidence with the CT. Sometimes X-rays are a little more difficult to discern. Now I'm going to look at a 3 case Dr. Dreyer submitted as well. And again, we've got several of these multi-level cases, and the statistics in the literature don't always support good outcomes. Once you get beyond 2 and 3 level, the fusion rates precipitously drop off. Yet with this case, maybe you could comment on what you see here at this early time point.
Yeah, I think we're seeing the same phenomenon we saw with the last case. We see the dense material, which is the magnetos, within the center window of these interbody implants. But you see bone up on those sagittal views in the anterior portion of that porous peak in front of the window, and the posterior portion of that porous peak behind the window. And then if you look at the coronals, the same thing. You can see bone to either side of that window growing through these implants.
Looking at the bone not only emanating from the graft window, but you can actually see it around the implant as well. And that was
Right.
quite encouraging in terms
And
of.
then at the bone implant interface, things look good. You know, I'm not seeing erosion or subsidence.
Alex, perhaps you could comment on Dr. Foley on the cases that Kevin has just shared with us.
So this case illustrates exactly why I like an invisible implant. Number one, the surgical technical attributes are perfect. Kevin has contoured the implants appropriately. He's left the subcondylar bone on. He's symmetrically placed the implant, and it's easy to do because the implant is designed that it has 2 tantalum beads in one portion, one in the other portion in terms of the anterior posterior dimension of the implant. So you can see that the cage has been perfectly placed. And if you look at the inserter, it gives you the opportunity to center it correctly. So, and then Kevin places his anterior cervical plate exactly the way you have to. So number one, congratulations to you, Kevin, for doing such an exquisite surgical job in placing this implant. Now you can see, look at the color of the vertebral body above and below, and look at the implant. You can see right through the implant. You have a perfect corridor. So over time, and you can see the size of the implant, it really maximizes the surface. So number one, that prosthesis is going to minimize any type of settling. And you can envision in the future if the FDA allows the center portion of the graft itself not to be hollow, and you have a fully 3D lattice in a diamond shape throughout to give it even more end plate support, that you wouldn't need any bone graft filler. But this is perfect. Now I can be comfortable. And if you look at this slide again, well-placed graft, and you could see at the 3-month part, you start to see that the ingrowth of the bone, you see the dissolution of the vertebral body end plate. In the picture on the left, you could see the end plates. In the picture on the right, you don't see the end plates as well, because now you're starting to get osteointegration with the implant itself and the filler. So well done, Kevin. Surgery looks great. You have maintenance of disc space height, and it looks like you're integrating the actual interbody spacer with the vertebral host bone.
So this is the other implant, and same implant, but same technique using autograft and local bone. Early responses. I don't know if you want to say something about this as well before we get to the CTs.
Yeah, I love Kevin's technique of taking local bone. He uses the burr. He catches the bone fragments and the dust, and then he sort of imprints it within the interbody space in the center of it. And it looks like a solid piece of cancellous bone, which is a great way of doing it, because you're using the osteogenic potential of the patient's own bone. And then you add it to the osteoconductive features of this interbody implant where cells love. And then you place it in, again, another technically perfect procedure as evidenced by the lateral x-rays. There's this superimposition of the different anterior and posterior borders of the graft itself. Here's another example on the following slide of a single level fusion, and you can see over time, 3 months, you start to see blurring of the respective end plates that are opposed to the interbody spacer, and a well-positioned graft. So hats off to this surgeon. Another well-done surgical procedure.
Yeah, Alex, if you could comment on the benefit of knowing that that bone is infiltrating at this early 3-month time period, what that means for you as a surgeon in terms of your confidence of osseointegration and ultimately fusion.
As a surgeon doing cervical spine surgery, you want to have your interbody spacer be sticky and stable at 6 weeks. And you can see on this 3-month x-ray, you're starting to see blurring of the end plates. You don't see any demarcation where there's evidence of non-healing. So you have a stable implant. And again, you don't need your plate after 6 weeks. In the future, possibly we can place this implant without an anterocervical plate. So with this, we have the ability to visualize the healing process. Unlike a titanium implant, we may have a difficult time seeing it.
Yes. And so here's the next case, 3-level case, again, 6-week time period. Earlier, your thoughts about this case?
So again, another great case. 3 levels are challenging. And you saw from the clinical series that there's 4-level cases, and believe it or not, 2 patients with a 5-level. So and where I see a challenge is I see a challenge on the inferior graft host junction, and you can see it's nice and stable. You don't see any kyphosing, you don't see any significant settling. So that this is a well-done procedure. And again, the surgeon has the confidence to be able to look at a lateral x-ray and say, yes, I can see what's going on. I'm confident that I have healing. And if you have non-healing, you can see it early. So if you have to intervene from a posterior approach to provide further stabilization, you have that option.
So as we're wrapping up all this wonderful information, wanted to open the floor for some question and answers. And one of the most common questions, there's a lot of PEEK out on the market. There's a lot of porous PEEK that has been generated through compression molding and other methods. The question is to the faculty here is, given the data that we've covered, the preliminary compelling clinical results that we see thus far, what about porous PEEK, or when you first heard of our Inspire porous PEEK, where maybe some of your inherent reservations, concerns, things you would want to see, and now having stepped through the science, the engineering, and the biology, as well as the clinical, what would you like to say about what you feel better about, what has helped convince you, or at least got your attention that we're moving in the right direction on this implant technology? Barbara, could you comment on that?
Sure. Thanks for the opportunity. When I think about materials that have to integrate with biology, I'm always thinking about structure, because the structure in biology is very real. when bone is forming, it's a highly vascularized tissue. No bone cell is ever living further than 100 microns from a blood vessel. So in some of the earlier versions of porous peak, what I would see when I would look at them is bone growing in for sure, and there was definitely a mechanical interlock, but always at the base of the bone between the growing in bone and the biomaterial, there was always fibrous connective tissue, which is the default pathway for bone marrow stromal cells or mesenchymal stem cells. For many of you remember the mesenchymal stem cells, they can differentiate into bone forming cells, but they can just as easily differentiate into fibroblasts if they don't have an environment that supports them being an osteoblast. And an osteoblast has an insane need for oxygen. So what I thought as I looked at this new structure was that there are no dead ends. There's no reason, particularly when you realize all that VEGF is being produced, there's no reason for the vasculature not to grow all the way through. And because it's there, it's providing the nutrients, the oxygen and the nutrients to the newly forming bone. And that's special. To me, that says that structure, that open structure, that trabecular bone-like structure is conducive to the ingrowth and through growth of bone, something that we haven't seen before. Thanks.
Thank you, Barbara. Kevin, if you could comment on your thoughts.
Well, you've made peak bone friendly, Eric. You know, the knock on peak is that it's not bone friendly, right? You're going to have fibrous tissue at the peak bone interface, and only grow bone through whatever graft window you have. This material, I think, demonstrates the exact opposite. This material likes bone, and bone seems to like this material.
So we hope to see a lot more of these, and this has been a phenomenal conversation, very insightful. This is exciting as a material scientist, seeing a culmination of decades of research and development effort. And really the 3D printing enabled us to be able to create this structure and this scaffold. And so as we kind of wrap up this conversation, there are some questions. And what we often get is, have we hacked peak? And in some ways, that may be the parlance of contemporary generation thinking about hacking things. We clearly have changed those properties. My question to the panel is, given your original perceptions about peak, what have you heard in this conversation, in this discussion, that has changed or altered your perspective, and you now think about peak or this type of 3D printed porous peak going forward?
So I've always been a fan of peak. So I used peak in the past in an off-label manner where I placed a bone morphogenic protein within the substance of it. So I used the biomaterial within the cage itself to help me heal. That was an off-label use because BMP has only been approved inside of a tapered metallic cage. And then over time, in the last several years, different industry partners have found that if you change the surface structure of a peak case, you allow bone to adhere to it and avoid the fibrous membrane. And now with HA infused throughout the entire implant, covering everything in a nano-sized particles that cells like to adhere to, I think you've taken what I believe is a great interbody option, and you made it a perfect interbody option due to the biomaterial.
Outstanding. So finally, to wrap this up, we at Curiteva, again, are committed to doing everything we can to advance this technology and meet the needs of clinicians for their patients. So I mentioned that we have a clinical registry to collect data and outcome measures for every one of the patients that we've collected. We hope to get this in the literature, and looking at CTs at 3 or 6 months. We certainly have future lumbar implants that are in development and testing, and certainly be evaluated clinically. What else could Curiteva do from your perspective to help surgeons make a easy choice about selecting this material and this implant?
All you have to do is what Curiteva is doing now. Keep a registry going, have physicians report their outcomes once they get IRB approval, keep a radiographic archive, and show the populace that this type of biomaterial, this type of structure, this type of superstructure with the coating is effective. And that's all you need to do. And then you'll convince people that this type of biomaterial is the way to go.
I'd like to ask Kevin his thoughts.
Well, we need longer term follow-up, right? No doubt about it. And a registry is very important here. You guys need to be following the patients and understanding how they're doing long term. You know, so far so good, right? But we need more data. I think the basic science from Dr. Boyan and others is incredibly helpful to help demonstrate that what we think is going on biologically in the humans, because we can't sacrifice the humans, is indeed going on biologically in the animals. And then I think that kind of experience can lead to some really interesting things to do with this material. For example, I'm not sure you need the graft window here, right? What I'm seeing so far is I'm seeing bone grow through this material. And so why have the graft window? That's an area in our interbody implants that doesn't immediately load bear very well. And we know that if we can spread the load over a greater surface, right, reduce the stress on those end plates in that fashion, that alone should diminish subsidence. And I think an added feature is that you may reduce the need for the volume of biologic material you might otherwise need. And I also think that the ability to make implants in a solid fashion out of this material can lead to some really interesting designs. If this material proves to be as good as we think it is, then I would want every interbody implant I use to be made of this material.
So that's a wonderful segue as we begin to wrap this conversation up. The nice thing about a 3D printer is the presumption you can print anything that you want.
Right.
And we're indeed, because we built our own machine and have a tremendous amount of organic knowledge in-house in terms of how to print different constructs. This slide represents all the things that we've actually been able to print on our printer. You can see PLIF, T-lifts. We've got larger implants. We're in the process of printing standalone ALIF. You see some plates that we've played around with there on the right. And this biologic incorporation, if you will, we can print different pore structures. And indeed, structure does drive biology. And in those lower middle sections, you can see tibial plateau and then Cotton and Evans foot and ankle wedges. Yep. I think, anywhere bone or a physiologic tissue needs to incorporate in a friendly or physiologic manner with an implant, that this is an enabling technology that will facilitate that. Maybe as we look ahead to lumbar implants, standalone and expandables and other things like that. That's all the time we have this evening for questions. And with that, I would like to thank the outstanding faculty for their time away from busy surgeries and for their outstanding input. I want to thank you, Barbara, Alex, and Kevin. It was really remarkable. And I'd also like to thank our surgeon pioneers. We have had a quick adoption and very keen interest in this implant technology. So I want to thank the surgeon pioneers for their early work. And importantly, some of our industrial partners I'd like to thank, researchers and industry partners at ProMimic, at Evonik, the provider of the filament, at MICRA from a regulatory guidance standpoint and empirical testing, Bill Walsh down in Australia. I want to thank all of those people. It takes a team to make this happen, and certainly the Curiteva team that's been working diligently on this technology. And last but not least, to our listeners, without you, we don't have the audience that we want to get this message out there so people understand that, as hopefully I build in the beginning, that this truly is living up to a revolutionary technology, data, and structure that we haven't seen before. And so look for more 3D printed trabecular lattice and clinical outcomes. We hope to see you at NASS, which is coming in October, and look for more to come from the team at Curiteva. Again, thank you for your attention tonight. Have a good evening.